over to you.
Okay, thank you, Andre. Well, welcome everyone to the Animal Interfaith Alliance's, AIA's, very first webinar. My name is Barbara Gardner and I'm a director of AIA. We're delighted that you could join us for what I believe is one of the most critically important topics of our time. What lessons can be learned from the coronavirus pandemic? I'm honoured to have Dr. Andre Menache here to present the webinar and for it to be introduced by our president, Dr. Richard Ryder, a founder of and a tower of the modern animal welfare movement. Dr. Andre Menache is our European representative and our science advisor and also a patron. So first I'd like to introduce the Animal Interfaith Alliance, or AIA, for those who are new to us. AIA's mission is to create a united voice from all of the world's faiths to bring about the compassionate treatment of animals. It is a unique alliance of faith-based animal advocacy organisations which represent all the major faiths. It speaks out on the moral treatment of animals by drawing on the combined wisdoms of all faiths, and it promotes social harmony by bringing the faith groups together on an issue they all share a concern for. AIA's activities include animal advocacy through its letter writing campaigns and education through its magazine, Animal Spirit, and social media. AIA currently has 17 member organisations, which you'll find listed on the website. AIA has recently written to the United Nations calling on them to take decisive action to prevent further pandemics. So now I'd like to briefly introduce tonight's webinar. There will be a 5-minute introduction by our president, Dr. Richard Ryder, which will be followed by Dr. Andre Menache's presentation, What Lessons Can Be Learned from the Coronavirus Pandemic. This will last for approximately half an hour and will be followed by a Q&A session. And we hope to end the session at around 10 p.m. Please do feel welcome to ask questions using the Zoom Q&A function during the presentation. I'll collect these and put them to Andre after the presentation. Finally, please note that the webinar is being recorded and can be watched and shared later on YouTube. I'd now like to introduce Dr. Richard Ryder. Richard is an ethicist who read psychology at Cambridge University, where he studied animal behaviour and obtained his PhD. He worked for many years as a clinical psychologist and psychotherapist in Oxford and was Mellon Professor at Tulane University. He became a key figure in the animal rights revival of the 1970s, coining the word speciesism and playing a leading role in its rise to international and political recognition. He was also director of the political animal lobby, PAL. He's currently the president of the National Society of the RSPCA and has been its chairman. His books include Victims of Science, Animal Revolution, The Political Animal, and Species, Painism and Happiness. I would now like to hand you over to Richard, who will give tonight's introduction to the webinar and to Andre.
Well, thank you very much, Barbara. Barbara's so modest that she fails to mention that she's also the founder of AIA, which is an organization, as you've heard, that brings together people from many different faith-based groups in their concern for animals. She's also, by the way, the national treasurer of the RSPCA. AIA was one of the earliest animal protection organizations to respond to the COVID-19 crisis by writing to the United Nations and the WHO in March 2020, calling for radical changes in the way humans deal with animals in factory farms and in live markets. AIA said human cruelty causes pandemics. AIA has not, however, fallen into the trap of making it sound as though we believe either A, that the virus is somehow the animal's fault, or alternatively, B, that the pandemic is God's punishment for human cruelty to animals. AIA has, I know, maintained its scientific and rational approach throughout. We have to bear in mind the current political situation with our government. They've made half a dozen promises that they have not yet kept on one, an Animal Protection Commission, which is an idea that some of us have been pushing for 20 or 30 years. Secondly, I mean, as a permanent body, as a permanent commission, not as a one-off commission. Secondly, they've made promises about American imports at lower animal welfare standards. Thirdly, they've got the agriculture bill. Fourth, the sentience or sentiency bill. Fifth, live exports. And sixth, sentencing. So there's a lot of things that they haven't yet resolved. They've made promises about them, but they haven't yet done anything. The questions which I hope we'll discuss today will include our attempt to, one, define our COVID-19 campaign objectives, for example, a ban on wildlife markets worldwide, or live markets to be banned. Secondly, to identify those bodies and individuals we should be targeting, for example, the United Nations, the EU, the OIE, Boris Johnson, WHO, G7, China, even Donald Trump. And three, some campaigning methods that we might use, for example, letters, emails, protests, head-to-head meetings, BBC, targeting BBC and other television and radio channels, the internet, Daily Mail, etc., etc., etc. I am incidentally too old for the internet, but I look forward to hearing the suggestions of others. Now, today we are honored to be addressed by the distinguished scientist, Dr. Andre Menache. We're lucky to have such an expert on zoonoses to address us on the scientific aspects of the case. Dr. Menache is AIA's European representative and scientific advisor. He is a veterinarian who advises extensively on animal welfare matters. Dr. Menache is scientific consultant to and former director of Antidote Europe, based in France. He's been president of Doctors and Lawyers for Responsible Medicine in the UK and general manager of the Federation of Animal Protection Societies in Israel. He's a patron of Quaker Concern for Animals and provides scientific support to several grassroots organizations. He's also a patron of AIA. Dr. Andre Menache.
Thank you very much, Richard. Thank you very much, Barbara, for that very warm introduction. Barbara, can you hear me? Barbara, can you hear me?
Yes.
Just let me know, is my voice okay? Not too loud, not too soft?
Sounds perfect, Andre.
Okay, so thank you again so much for that very warm introduction. I'm just going to dive straight into the presentation. So here we go. So lesson number one. And I very much like this quote from Chris Packham, that the virus has taught us that we are part of nature, not above it. There's a lot of wisdom in there. Now, for those of you who are not familiar with the COVID-19, with this coronavirus, it looks like this, but it's much, much smaller. For those of you who are scientists, yes, it's a single-stranded RNA virus. And it's called a coronavirus because when it's seen under the electron microscope, it has a sort of a crown on it. Looks like it's wearing a crown. Now, the spikes that you can see on the surface, that is the equivalent to the key to unlock the cells, to let it inside the cells, to let it get inside the human body. So that's just to simplify what you can see there. So Richard mentioned the word zoonoses before. Zoonoses are diseases transmitted either from animals to people or people to animals. Usually they refer to animals to people more than the other way around. And they can include bacteria, viruses, all sorts of parasites. So that's the word zoonoses. Now, where did this coronavirus come from? So there are two basic hypotheses. The first hypothesis is that it came from either a bat or a pangolin. Now, if you look at the structure of the coronavirus in bats, it's not identical to the coronavirus that's infecting humans at the moment. So what it could mean is that the virus had to mutate before it could infect people. And if you look at the structure of the coronavirus that is found in pangolins, I believe that resembles the human coronavirus even less. Now, what I can't get my head around is how on earth this virus could have been transmitted from bats to pangolins to humans, because I don't think that bats and pangolins meet up very much for coffee. So that would be that I find scientists have to explain to us, unless of course it's during wet markets, the sale of these animals at wet markets. So that's the first hypothesis, is that this virus, this coronavirus, this disease has come from bats and or pangolins. The second hypothesis is that this virus is actually originates in a research laboratory. Now, we do know that in China, bats are studied quite extensively in laboratories, and it's possible that this virus could have emerged from a laboratory. We don't expect the Chinese government to come out and say, yes, you know, we are responsible for creating this pandemic, but so we're not sure yet where this virus comes from, but it's one of these two. It's either from wild animals directly or from medical research in a laboratory, and then somehow that virus got out of the laboratory. So another lesson that the coronavirus has taught us is that we should be very careful in the way that we deal with wild animal species. In other words, animals that we normally would not come into contact with. We have evolved over millions of years. Bats have evolved over millions of years, and we all have our own viruses and our own bacteria. And, you know, normally, you know, bats are quite happy in their caves and they do their thing. But there are certain human activities that actually favor the creation of dangerous new zoonotic diseases. And we'll go through these one by one. So you'll see the bullet points: keeping wild animals in captivity, close contact between people and wild animals, overcrowding and unhygienic conditions, the destruction of ecosystems, and the wildlife trade. So the first one is keeping animals in captivity. A lot of ducks and bird species actually contain viruses like flu viruses, the human flu virus. And that virus does not bother the bird. And normally in water, that virus will pass out of the bird through the water into another bird in the water, and they live in harmony, no harm done. The problem comes when people start to take these waterfowl out of the water and put them in cages on the land, on dry land. And then the virus, which is very clever, viruses are very, very clever. They might, for example, mutate because they can no longer be transmitted through the water. They might then mutate so as to enable them to be transmitted through the air, so via droplets, for example. So that is one example of how humans have created a virus that was normally harmless in the water, passing from one bird to another. We've now caused this virus to mutate and become air transmissible. Another factor that increases the risk of a virus turning nasty is keeping different animal species in unhygienic conditions. Now, I'm not talking about sanctuaries where you find different animal species happily together, where the conditions are hygienic, there's lots of room for the animals to move around. I'm talking about unhygienic conditions where you have, as you can see in this photograph, you've got a pig, you've got some chickens, you've got a dog on the right-hand side. So those are conditions where, again, viruses can mutate, can jump from one species to another, and in doing so, they mutate. And you create viruses that are part pig, part avian, and they exist, they exist. Another practice, of course, is intensive farming practices. Now, it's not in the interest of a virus to kill its host, because in nature, if we look at the picture on the right-hand side, in nature, animals are normally spread out. They've got plenty of room to move around, and viruses have evolved over millions of years. It's not in their interest to kill their hosts, because if they kill their host, that animal will die. And if another animal is far away from the one that's died, then that virus is actually committing suicide. So most viruses are actually not, it's not in their interest to kill the host. However, when we provide a situation like the photograph on the left, then actually the virus has nothing to lose by becoming far more pathogenic, far more virulent, far more dangerous. Because there, if it kills its host, it's already moved on to the next and the next and the next. So again, this is an example of how human practices, in this case intensive farming, creates viruses that are far more virulent than they will normally be in nature. So here we have a summary where we've created a vicious circle. We've created a virus that is air transmitted. We've created a virus that's part pig, part bird. And through intensive farming practices, we've created a virus that's become far more dangerous, far more virulent. And then, of course, the people who work in these establishments are the first people who will be infected once the virus mutates and adapts itself to a human host. So there you have the sort of the full cycle, beginning on the left and ending on the right. Now, of course, another source of animal viruses are wet markets. Unfortunately, this is a practice in some cultures more than in others, but these are ideal conditions for viruses that we would never normally come into contact in nature. We here become very exposed to them. So this is definitely something of great concern. The next issue is the destruction of ecosystems, because what happens is when ecosystems are destroyed, the animals get pushed closer and closer together because there's less habitat. So they are more at risk of picking up viruses from each other. But also, it means that they are closer and closer to human habitation. And so that we also increase the risk of wild animals entering human habitations. So that's only part of the story. There's obviously much more to it than the destruction of ecosystems, but I'm just focusing on viruses at the moment. And then finally, the legal and illegal wildlife trade. I say legal and illegal because you can just as easily pick up salmonella from a reptile bought in a shop as you can from a reptile that was caught in the wild illegally. And I've put on the website, the website below, the emergentdisease.org, because it's an excellent website, excellent resource, where you will discover some very disturbing facts. For example, the fact that 75% of all reptiles, especially snakes, that are purchased by people who think that they make great pets, 75% of them are dead within a year of purchase. And some of these animals live to 25 to 30 years old. So obviously people don't know how to look after these animals. So that in addition to the public health aspect, there's a huge welfare aspect to this. Another lesson is, and this comes back to the hypothesis number 2, that this virus could have come from a lab. Now, the Pirbright Institute in the UK is the highest level biosecurity laboratory that you can find, and it's called a P4, which is the same as the one in China, the Wuhan Institute, which is also P4. And yet we have an example in the UK in 2007 where a foot and mouth virus escaped from this establishment and caused some panic in nearby farms. So we can see that these P4 institutes are not watertight, and really should we have these institutions? Some of them are built very near cities. I mean, that is an accident waiting to happen. The only real reason we need biosecurity institutes is if we need to, for example, isolate and identify a pathogen in people or in animals. But then, you know, normally a P3 does the trick. We don't need to go up to P4. P4 is already getting a bit dodgy. P3 is sufficient, and of course we shouldn't build it. We shouldn't build these institutes. We shouldn't have them near human habitation. You know, put them on some little island somewhere in the middle of nowhere. Do not build these in human built-up areas. Now, just a word on our companion animals. Now, there's far more risk of us transmitting coronavirus to our pets than the other way around. Okay, the current scientific evidence suggests that the risk of us getting coronavirus, COVID-19, from our pets is about as likely as finding rocking horse droppings. So it's very, very unlikely. What has been done in some labs is, under laboratory conditions, cats have been infected, for example, with the coronavirus. Now, we don't know what dose they've been given. Perhaps they've been given enormous doses. And yes, they have transmitted it to other cats. But bear in mind that these are under laboratory conditions where the animals are normally very stressed. And once stressed, that would lower their immune systems. And we don't know what doses these animals were given. So really, our companion animals, based on current evidence, we really should have nothing to worry about. They have more risk of contracting the COVID virus from us than vice versa. Also, don't forget that they are not as susceptible to the virus as we are. Humans, the virus, the COVID-19 is really custom built for humans. It's mutated specifically for humans. Now let's just a word about medical research. So the COVID-19 caught a lot of scientists by surprise, because they had no animal model to study this disease. And so a lot of the research was actually human-based, was actually intelligent research. Unfortunately, now scientists are beginning to use animals to study the disease, although we have masses of human data. And of course, we are not 70-kilogram mice or 70-kilogram ferrets or 70-kilogram monkeys. So just a word about the role of animal research and testing. There's a lot of confusion here, because animal researchers will normally justify animal testing by saying that the law requires them to do it. They do it because the law requires. That is actually very disingenuous. Yes, if we look at the history of the legal requirement for animal testing, it goes back to the doctor's trial at Nuremberg on the left-hand side at the end of the Second World War. Because of the issue of human experimentation, it was decided there and then that before any testing is done on humans, one must test the drugs or the chemicals on animals. So that is a 70-year-old law. And unfortunately, the laws have basically stayed the same since then. The legal requirement for animal testing is the same today. It hasn't kept up to date with the science. And here comes the quote on the right-hand side. Now, James Gallagher, past director of research of Ledley Laboratories, is somebody within the pharmaceutical industry. And he stated in 1964 in the Journal of the American Medical Association that animal studies are done for legal reasons and not for scientific reasons. The predictive value for such studies for man is meaningless, which means our research may be meaningless. That is a very, very important quote, because I think that puts things into context. And the reason why the animal testing is done is because the law requires a rodent and a non-rodent species to be tested on before the pharmaceutical company proceeds to clinical trials on humans. So let's stop the witch hunt. So it's not the bats, it's not the pangolins that we should worry about. It's more our behavior. Okay, we are the cause of the problem. And either because we are eating wild caught meat and wild animals, which we shouldn't be, or else because we are performing research in laboratories, tinkering with viruses, and sooner or later there's going to be an accident, as was the case in Pirbright, and apparently as could have been the case in Wuhan in China. Just a word about unsafe science while we're on the subject. Xenotransplantation is the transplantation of organs from animals to people. And every few years, some researchers will bring up this idea that we can use animals as spare parts, either for hearts or kidneys. And it's just bad science. It's actually no different to wet markets, because what we're doing is we're introducing animal viruses directly into people. So that is very, very dangerous. And you cannot weed out all these viruses. You cannot sterilize a pig heart. You can sterilize a pig heart valve, yes, a valve, but you cannot sterilize a pig heart. And so what you're doing is when you transplant a pig heart into a person, you're going to transplant viruses known and unknown, and even prions. Now, prions are things that cause mad cow disease, for example. And pigs have their own prions. They have the gene that makes the protein to make a prion, just as humans have the gene to make the protein that makes a prion. It's on our chromosome number 20. So you can't get away from these things. You cannot, there is no way that you can completely exclude the risk of transmitting viruses from, or prions from animals to people. And then I think the COVID-19 is also an opportunity to talk about finding safer vaccines. Currently, there are about 148 vaccine candidates, and about 17 are in clinical trials in humans at the moment. And some of these vaccines, including the one manufactured, provided by Oxford University researchers, contains a genetically modified chimpanzee virus. Now, I don't see the difference between wet markets and the risks of transmitting animal viruses to people by eating animals and injecting modified animal viruses into people. So I think we must really, it's time to look at the way we produce vaccines. I mean, most people have no clue about how vaccines are made. The fact that a lot of the cell cultures used in vaccines that are used for people, destined for humans, contain animal cells. Okay, duck embryo, monkey kidney, and occasionally aborted human fetus. So we can use our own cells. There's nothing safer than using our own cells to manufacture custom-made, individualized vaccinations. That's just beginning now. It's just beginning, but that's a subject for a few webinars. In fact, it's a vast subject. So I'm going to go on to this example. So this is a an in vitro, a very sophisticated in vitro simulation of a human immune system that uses human cells, very sophisticated, and this is the way to go. And yes, more and more pharmaceutical companies are going down this route, but at the end of the day, they will say, but we still have to test on animals because the law, that's what the law says. And here we see why we need to change the law, why the law needs to say, tested on this sort of human-based technology and stop the animal testing. So I think what the overall message is, it's time to harmonize with nature and stop fighting nature, stop trying to beat nature, stop trying to overpower nature. And some just interesting facts and figures. There are more bacteria in our bodies than there are human cells. There are 10 times more viruses than bacteria in our intestines. And about 8% of human genetic material comes from a virus and not from our ancestors. It just shows you how clever viruses are. They're going to preserve themselves. They are going to outlast humanity, that's for sure. So if you can't beat them, join them. So finally, be the change you want to see in the world. In other words, don't only expect the authorities to make the changes. We as individuals, whether it's cutting down on plastic, whether it's cutting down on meat, whether it's cutting down on driving in a car, we can make so many changes at the individual level. And you cannot believe the power of the consumer. Consumer power is what is going to change industry's habits. It's what's going to change cultural habits and so forth. And in concluding, I would like to ask Richard Ryder for some words of wisdom. Thank you, Richard.
Goodness me, you've taken me by surprise. I'm very impressed by what you've just said. And I've learned a great deal because I find the whole issue quite complicated and quite intimidating. I tend to think in terms of how we can get results and how we can change the law, how we can get governments to act. And one of the element of any good campaign has to be, of course, the inclusion of scientists. And people like you, Andre, are a tremendous asset to the movement. You manage to explain very complicated ideas with great clarity, and that's wonderful. Thank you very much indeed. I haven't prepared any particular words of wisdom, other than the few I said at the beginning, so I don't want to repeat myself. I just would like to listen to the questions that people have to ask and see what the answers are. I'm always thinking in terms of an end result, however. I would like to get something happening. We need to follow up our initial approaches to the United Nations. Maybe there are other bodies that should be involved, but surely whether or not there's a vaccine by the end of the year, we ought to be thinking in terms of changing our attitude and treatment of animals. And we ought to be, whatever happens to the science and whether or not we've got a vaccine by the end of the year, we ought to have some agreement internationally among politicians as to what needs to change in our own human behavior and in our treatment of the other species. Can I hand you back now, please?
Hello, well, thank you very much, Andre. That was a fantastic presentation, full of information that I think all of us, will feel we've learnt so much from. I haven't got very many questions that have come in, so if anybody still wants to ask some questions, please feel that they can put it through on the Q&A screen so that we can take them. The first two I have, the The first one is, what is the issue with the outbreaks in the meat processing plants? So that's really, I imagine, to do with why are we getting outbreaks of COVID-19 in meat processing plants as opposed to anywhere else, Andre?
Well, I think one of the problems in the in abattoirs is the close contact between workers. So that could be one issue, because when the COVID-19 pandemic began, most governments put a stop to any gatherings of groups of people who were, you know, more than 2 or 3 people was a no-no. Everybody had to wear masks, etc., etc., etc. Now that in an abattoir situation isn't possible. You've got lots of people walking around, very stressed out people. So I think perhaps abattoir workers may actually be more susceptible than people who just have a normal day job or who work from home, obviously. Because, by the way, most of these viruses actually don't kill. Most of these viruses, I'm talking about the COVID-19, this is the evidence that we have today. Most of the people who have passed away, who die with the virus, not because of the virus. This is even true for the Spanish flu of 1918 that killed about 50 million people. It seems that the virus weakens the immune system, or else it causes other complications, but it's not the virus itself directly that is responsible. So I think we have to look at the bigger picture. In other words, who are the people that are most susceptible to getting the virus, and why is that so? We know that, for example, in the case of COVID-19, people who already have high blood pressure, people who are already obese, people who already have diabetes, people who already have respiratory problem, they are more susceptible. So I think there's a huge, we must put emphasis on prevention. I haven't seen this in any government public policy. Everybody's spending millions and millions of euros or dollars to try and manufacture a vaccine, but nobody's dealing with the root cause. We're not seeing the same amount of political will, if you like, on banning wet markets, on banning the consumption of wild meat. That would be a fantastic way to increase prevention. So I hope I've answered that part of the question. And then the second part of the question is, what is the process of vaccine manufacture re-animal testing model? Well, I think I've partly answered that already. In other words, the animals are used because of a legal requirement that says, yes, you must use the animal beforehand. What the researchers then do is they try and find an animal that suits them more than anything else, because you know, we're not ferrets, we're not mice, we're not monkeys. Whatever happens in those species is species specific. No animal can predict what will happen in a human being in terms of drug development, in terms of disease development. So unfortunately, that is a concept that belongs to the 19th century. And sadly, it's still the dominant or the predominant paradigm in biomedical research today. So that has to change if we want to move forward. So I think that's the short answer to that part of the question.
Okay, thank you very much, Andre. We've got a few more that have come in just in the last couple of minutes. One is regarding the risks of using animal parts for transplant into humans. Could you say more about what might happen with some specific examples of cases?
Sure. So if we look at, for example, I could use the example of the early polio vaccine, because the early polio vaccine used between 1955 and 1960 was originated using chimpanzee or monkey kidney cell culture. and some of those kidneys were naturally infected with a monkey virus called SV40, which stands for simian virus 40. Now, the SV40 virus is the most potent cancer-causing virus known to science. And it was only after about 8 years of use, so in other words, millions and millions of people had already been vaccinated, some of them with this contaminated vaccine, that scientists started noticing unusual and rare sorts of cancer in people, and they actually found bits of the virus DNA in those tumors, in those cancers. So there's very strong evidence that SV40 virus contaminating the monkey cultures contaminated people. That's one example. Another example is actually in the UK. Around the year 2000, there was a huge public outcry in the wake of the mad cow disease outbreak, because it was discovered that UK cows, or rather UK fetal calf serum from cows, was being used to manufacture polio vaccine in the UK. And of course, once the public outcry was made public, and the government quickly stopped using those vaccines and sort of tried to calm everybody down. But it's sad that our health authorities do these things behind our backs, and it's only because of people who sound the alarm, or concerned parents or whatever, that these things come to light. So in the case of if we use, for example, a pig virus, a pig heart rather, a living pig heart, it's not the same as using a pig heart valve. A pig heart valve, as I said earlier, is something that you can thoroughly sterilize, and even so, it only lasts about 10 years in the patient. So you can replace, or you can use an alternative to a pig heart valve in the form of a synthetic or a cadaveric, a donated human heart valve, which will last about 20 years. So even in the case of the pig heart valve, there's an alternative. But if we're talking about an entire pig heart, well, as I said earlier in my slides, part of our DNA is actually viral DNA. We have virus DNA within our own genomes, as does the pig. So there's no way that you can completely clear all these genes out of the pig genome. And even if you could, there's still prions. There's other things that we don't even know about. We don't know that exist. And we still haven't overcome the problem of rejection. Rejection is still a serious problem when it comes to organ transplantation. So I hope I've sufficiently answered that question.
Yes, thank you, Andre. We've got some fairly topical ones now. One around Brexit. What risks does Brexit pose for relaxation of standards or disconnection from European agreements?
Okay, so I've been out of the UK long enough not to be up to speed to answer that question. All I can say is that you know, the UK has a very long track record of animal welfare, and I think this is an opportunity for the UK, once it's sort of free of the shackles of the EU, to actually go above and beyond the minimum requirements that the EU has imposed on other EU member states. So actually, you know, there are a lot of, I know there's a lot of bad blood and pros and cons of Brexit, but I think here we have an example where the UK can lead the way, if you like, and go above and beyond what the EU requires.
Okay, thank you very much. Quite a fundamental question here. Are all virus pandemics pretty much down to us mistreating animals in some way?
Well, I think the short answer is yes, because you know, nature has put up, if you like, red lines or barriers. You know, we should not be eating bats. We should not be eating pangolins, because you know, in the greater scheme of things, these animals should be wild roaming. It's not easy for humans to come into contact with these animals, and then for some reason we just decide, you know, we want wild caught meat, we want bush meat, we want fancy exotic animals, and I think we are disturbing the harmony with nature here. And I think that it's so clear when today there's more and more research about how important it is to keep our gut bacteria happy, because our gut bacteria have a huge impact on our immune system and even on our mood. So, you know, we're only just beginning to discover and to understand our relationship with the animal kingdom, with microorganisms, with bacteria, with viruses, and so forth. And there's no way that we'll ever beat them or be stronger. And we have to, I think, come away with a more pacific and more harmonious approach to the way we treat this planet. And I think that is what the COVID-19 is, I think, is trying to teach us. One of the lessons, anyway.
Indeed. Okay, so moving away from the wildlife markets and onto intensive farming. And one of the questions is that in view of the origin of almost all the pandemics, is it not only time to call for an end to wildlife markets, but also to call an end to intensive farming of animals and to stop eating creatures entirely?
I think absolutely that's a fundamental key. And I think there are two approaches. The one approach obviously is to lobby parliamentarians and try and find legal ways to stop this. But the other is also at the level of the individual. To me, there is no more powerful example than the example of the individual. So we must all sort of begin with our own plates and with our own homes and with our own families, because that's where we can really do something. We certainly cannot rely on the authorities and hope that, you know, in 20 or 30 years time they're going to change the law. So in the meantime, we have to do things. Yes, we can certainly use modern technology like social networking. I think that's hugely important. So I'm very much of the opinion that we have to work on two levels. If you like, the parliamentary lobbying, but also very much what we do as individuals. And it's more than just stopping to eat meat. I mean, for example, if you stop using plastic, that's going to have a positive impact on the destruction of ecosystems. So it's actually all tied up. It's all linked in sort of one whole packet.
Okay, I think one of the things that has struck me through this pandemic is that it's all very well for us to do our own thing, which I think nearly all the attendees here will have done, you know, by avoiding cruelty to animals in all sorts of ways. But unless we communicate this to others and campaign on it, we're still all going to be affected by things like this pandemic. So we've got to actually go that step further and not just change our own way of life, but try and communicate the problems to others and influence change. I don't know how you feel about that, Andre.
I totally agree, and I think we're very fortunate today to have social networking, because we're not totally dependent on the mainstream media. Although I have to say that, you know, mainstream media cannot ignore us all of the time, because occasionally, yes, they do say, well, it's actually okay not to eat meat, and etc., etc. So yes, I think let's use all the tools at our disposal to communicate these messages, and education, of course, is key. You know, education beginning as early an age as possible. It would be nice if animal rights or animal welfare organizations could talk to children, could get into schools as easily as some of the dairy and the meat industry lobbyists do.
Yes, that's a very good point indeed, Andre. Just moving on to something a bit more biological, it's about viruses, and somebody has asked, why do they exist peacefully in animals and become more virulent when we tamper with them or they transfer to humans?
Well, I mean, viruses have evolved over millions of years, and viruses were probably on the planet way, way, way before mammals. So they are here. They were here first, if you like, and they have very cleverly learned to live in harmony with whatever evolved. So it might have been dinosaurs, and then it might have been reptiles, and then mammals came along. So 8% of our DNA is viral DNA. So they've learned to evolve and coexist with us. And the example I use to illustrate why viruses turn nasty when they find themselves in a strange host is if you let kids do what they want in a playground, and you let them and they do naughty things or they start breaking things, and you just let them run wild. So I think this is what viruses do, is they find themselves in a strange environment, and they just they do what they do best, and that is invade cells. And normally they live in total harmony with their hosts. They only bother their hosts or invade their hosts when their hosts, if you like, the immune system of the host drops. So when their host becomes ill, they will perhaps then opportunistically cause some pathology. But otherwise, it's a story of harmony. They've evolved over millions of years, and it's us that are creating these very unnatural situations and breaking down, crossing nature's red lines and breaking down natural barriers.
Thank you. But we come on to a question about moving away from animal testing to human testing. And the person asks the question of what level of funding would be required to advance human testing rather than animal testing. Are we talking millions or billions? Or maybe we're saving money?
Well, unfortunately, at the moment, most of the funding that goes towards biomedical research goes towards animal-based research, because that is the current paradigm of biomedical research. It's still the animal model. I have spoken to researchers who have told me that it's easier for them to get authority to experiment on 100 rats or 100 mice than to get permission to use discarded human surgical waste that is going to be incinerated. So there's a huge disconnect. So that's problem number one. Well, to answer the question, if the money that was used for animal research was put into human-based research, that could happen overnight. That could happen overnight, and we would see far more. We would see evidence-based medicine, because animal experimentation, unfortunately, is not evidence-based. So we would see a huge improvement in medical research, in the quality of animal research. There was an article that came out today that shows that 89% of preclinical research, including animal research, cannot be reproduced. In other words, another scientist, or sometimes even the same group of scientists, cannot reproduce their own animal studies. So, and this has been going on for years, for decades, and it's becoming very embarrassing to some in the animal research community, because they are at least open about it. And they are trying to improve the situation by saying, for heaven's sake, you know, improve your methodology. But of course, the scientific journals and the medical journals only make recommendations. There's no obligation for an animal researcher to improve their methodology. And even if they did improve their methodology, that would make the animal model no more relevant to human medicine than it is today. So as a veterinary surgeon, you know, I'm never going to test a drug intended for horses on parrots. And people get that straight away and they find it quite funny. And so I say, so why then test a drug destined for people on rats? Because we are not 70-kilogram rats. So I think the technology is there to replace animal testing tomorrow. What is missing is the political will. And also, to come back to what you said earlier, Barbara, it's communication. Because how do we communicate what we've been discussing this evening, which is, you know, 3/4 of an hour we've been discussing now. When you normally get, I mean, people like myself, for example, I might get 2-minute soundbite on radio or TV. And if I'm against an animal researcher, the animal researcher in 2 minutes will say, well, if it wasn't for animal testing and animal research, we wouldn't have vaccines, we wouldn't have antibiotics, we wouldn't have transplants. And that's it. I cannot undo all of that in 2 minutes. It's impossible. And the problem is that most of the public, most parliamentarians do not have a science background. Now, I'm not saying they all need to go and get a PhD in biology, but what I am saying is that at least let's hear the experts on both sides. So, for example, if we could have a science debate, and in other words, a parliamentary sponsored debate with experts, scientific experts on both sides, judged by a panel of independent experts, because unfortunately, most politicians do not have the science background to judge, to know who's telling the truth or who's putting forward the best arguments. So that, to me, is one way to get the science across. But at the end of the day, animal testing will stop, I believe, 10% because of the scientific argument that it doesn't work, and 90% because people will say, well, it's cruel, we're causing suffering, it's unnecessary suffering. And the animal research community has said for decades, we would love to use alternatives, but you know, at some point along the line, we have to use a whole living system. And there's no emotional argument to that. You have to reply with a scientific argument. And if you know the scientific argument, then the unnecessary suffering becomes simply unnecessary. It's not a necessary evil. So all that's left is the evil, not the necessary.
Yes, thank you, Andre. It's obviously a very biased and weighted system in favour of animal testing. And just before I go on to the next question, could I just ask you to remind us what the success rate is for animal testing and for non-animal testing methods?
Okay, so according to the Food and Drug Administration, which is the highest authority in the United States, they say that for every 10 drugs that successfully pass animal testing, 9 out of 10 will fail during human clinical trials, either because they are toxic or because they are ineffective in human patients. So that is a 90% failure rate. Now, in contrast to that, the regulatory authorities, like the FDA, will require today of a pharmaceutical company that wants to present a non-animal testing method, they will demand or expect that non-animal testing method has a predictive or success rate of predicting the outcome in humans in the range of 85 to 90%. So it's actually, it's almost the opposite of what animal testing does today. So we could say that if we had nothing better, if we had no non-animal methods available, it would still make sense to get rid of the animal testing, because 1 out of 10, a success rate of 1 in 10 is actually far less than 5 out of 10, which is the result that you would get from flipping a coin, from just guesswork. So that's how bad animal testing is. But of course, the public doesn't know this. And I think if the public knew this, there would be a huge outcry.
Okay, so even if the public didn't know it, surely, I mean, to me, it would seem the argument that you would have with the establishment was that an 85 to 90% success rate has got to be better than a 10% success rate. So how can they argue against that?
Okay, so the problem is that the regulatory authorities say to the pharmaceutical industry, we expect you to provide drugs that are safe and drugs that work in people. How you do that is up to you. If you want to provide us with animal data, that's fine. In fact, we're very comfortable with animal data because that's what we've worked with for decades. However, if you want to present us with human-based data based on human cell culture, human organ on a chip, etc., etc., then we're going to raise the bar. We're going to raise the bar because we think the public expect, something in the region of 85 to 90% success rate. The animal data we will continue to accept on historical grounds because we've got so much animal data. We haven't even validated it. Okay, animal models have never been validated, and this information that I'm quoting comes from a high-ranking authority from the FDA, who I met at a World Congress in the Netherlands in 1996. And I have this in an email from this person. So from the Office of Science of the Food and Drug Administration. So they clearly said to me, we accept the animal data because it's just been there for decades and decades. But the animal model has never been validated, scientifically validated. However, if you want to replace the animal testing with this wonderful new technology, which is far more relevant and reliable than animal testing, we're going to raise the bar. You have to provide data that is 85 to 90% predictive of human outcome.
So the scientists are being totally unscientific there, which actually brings us on to very nicely to the next question, which says, aren't a lot of animal experiments also about money? And don't we have to beat corporate propaganda? Aren't government grants awarded to research centres for animal experiments?
Unfortunately, yes, that's very true. It comes back to the point I made earlier, that the animal model is still the current paradigm in biomedical research today. It's much easier to get grants for animal testing than it is for example, human surgical waste that's going to be incinerated. So unfortunately, and of course, anything to do with prevention, it's very, very difficult to get funding for. So we have to, the whole system has to change. And the way I see it is that politicians need to be informed, the general public needs to be informed. And one way of doing this, one first step, would be a parliamentary debate. A parliamentary debate with experts on both sides, but the judging panel would not be politicians. It would be independent scientific specialists. So you'd have a specialist in complexity theory, you'd have a specialist in evolutionary biology, and so on and so on.
Great. Well, that sounds like a good way forward, a parliamentary debate. And maybe that's something that we could all work towards trying to get set up. Okay, so moving on, one of the questions is, well, we've got COVID-19, but what was COVID 1 to 18? Now, presumably COVID-19 is representative of the year 2019, but there have been, they might not have been called COVID, but there have been other viruses in the past, such as SARS. So what was they? And my added question to that would be, why didn't we learn the lessons from those and prevent COVID-19?
Okay, so COVID-19 is actually coronavirus, SARS coronavirus number 2. SARS coronavirus number 1 was a previous a pandemic, but it was I think just limited to 1 or 2 countries, 1 or 2 or 3 countries. So I'm not sure that there's 19 others or 18 others. And also there was MERS, M-E-R-S, which was also, I believe, a coronavirus. Yes, we've learned nothing from those pandemics. There was an attempt to make a vaccine, but the disease died out, petered out before a vaccine could be manufactured. And I think that most of the race to produce a vaccine, to find a vaccine now, is to save the embarrassment, because normally to develop a vaccine from scratch takes 10, 15, 20 years. And here we're seeing a vaccine that's going to be produced in 10 or 20 months. So there's no way we're going to know what the long-term side effects or even the medium-term side effects are. If people start dropping dead, then we'll say, okay, it's, let's stop. But unfortunately, if people don't drop dead, then the political reaction is very, very slow. So the reason that the governments have been so reactive to the COVID-19 is because people are dropping. And so even though more people die from air pollution than from COVID-19, you don't drop dead in the street from air pollution. It takes months or years, and you die probably in a hospital bed gasping for breath. And that's not, politicians can get away with that sort of thing. It's not, the pressure to alleviate air pollution is not as bad, because the whole thing around this is the fear of dying, dropping dead now. And that's why the money has been so forthcoming in the research for a vaccine. So the governments want to save themselves huge embarrassment in case the virus just dies out before the vaccine comes onto the market. And by the way, this virus is probably mutating. So it's a bit like the flu conundrum. You're going to produce a vaccine, but is it actually relevant to the mutated form of the virus? And then the second part of the answer to that question is that if you look at the 1918 pandemic, in other words, so-called Spanish flu, it was called by H1N1, probably of avian origin, or certainly with genes of avian, of bird origin in them. So already in 1918, we had a huge warning, a huge alarm of what happens when, well, this could be, for example, just keeping ducks or waterfowl on land. That could be enough. And also intensive poultry farming. That probably could be enough to have kicked this off. So already in 1918, we missed that. And that was 50 million people dying. And we haven't learned the lessons since then. I don't know if we're going to learn it now, but wait and see.
Yes, I imagine if COVID-19 peters out, we'll forget about it very quickly and not learn the lessons until the next one comes along. But it's interesting why have some of the other ones petered out, and this current COVID-19 continues to be so aggressive. What is so different about this one?
I think it's a question of creating the perfect storm. In other words, a lot of scientists have been saying for quite a while, it's not a question of if, but when the next pandemic comes around, and it's probably going to be worse. So you have perhaps, whether it's the wet markets or whether it's escape from the lab, you have a virus that's adapted itself to humans, and also it's gone through that cycle that I discussed at the beginning of my presentation, where it's been made transmissible by droplet form through the air, where it's become partly part bat, part bird, etc. And also it's become adapted to humans, but also it's become far more virulent because you know, when people live close together, as I say, the virus has got nothing to lose by killing its host, because it knows it's going to, you know, get into another host pretty quickly. Although we're not all equally susceptible to the COVID-19. People in good health generally have very little to fear. People in good health who've got a good immune system generally have very little to fear. By far, the most people who have died from COVID-19 are people with already pre-existing conditions, or, you know, some very, very unfortunate person whose genetic makeup just makes that person very highly susceptible. But that is the exception rather than the rule.
So isn't that just the same as seasonal flu?
It's the same as seasonal flu, except with a twist that this virus is passed on probably more quickly, and also has causes more complications, because seasonal flu doesn't cause you know, some of the pathologies that we've seen with COVID-19. So that's the difference. That COVID-19 has got a sort of a wider spectrum in terms of the complications that it causes, and also it seems to be more easily transmitted.
Okay, so changing the subject slightly, we've had one suggestion that we should request to our Prime Minister that we shouldn't eat meat in the same way that we should wear masks. So the Prime Minister should say no meat eating for a month. How much success do you think we'd have with that?
I really don't know. That's a $64,000 question. I don't have an answer.
Okay, so I think we've covered most of it. I'm just looking to see if there are any other questions coming in. Somebody's asked why they won't change the law so researchers are not bound to test animals. Is too much money involved? And is this the same reason for the meat and dairy industry, and why that's so extensive? It's all about money.
Well, money is certainly a huge issue here. As far as drug testing is concerned, it's much easier to get a drug onto the market and quicker to get a drug onto the market if you can use animal species, because the pharmaceutical manufacturer can pick and choose which rodent and which non-rodent species suits them best. I mean, I know of no other industry where this happens. So can you imagine if the pharmaceutical manufacturer was reduced to using only human-based methods? They would have far less room for maneuver. It would be far harder for them to manipulate their data. And that's why, I think that's one of the obvious reasons why they won't let go of the animal testing, because it just allows them to switch and change species and find the animal species that most suits them. Now, this doesn't only apply to the pharmaceutical industry, it applies very much to the chemical industry. And in the case of the chemical industry, the testing requirements are less strict than for the pharmaceutical industry, but the health consequences are just as devastating, because, you know, we're all exposed to pesticides and chemical cocktails, and babies born today have around 300 synthetic chemicals in their bodies that shouldn't be there. And, you know, if you take things like glyphosate, Roundup, pesticides, or bisphenol A, the plasticizer, these products have to be on the market for about 30 to 40 years before we see the negative health impacts on people. So in other words, the human body is incredibly resistant, you know, especially to cancer, most cancers. And so it will take about 20 to 30 to 40 years before the harmful effects of these chemicals show up in humans. And then you've got to go and take on the chemicals industry and prove that this particular pesticide that you've been exposed to as a farmer for years and years, that's caused your cancer. It's not easy to prove, really not easy, because we're exposed to so many chemicals. But the problem is that once a chemical gets on the market, in other words, once the manufacturer has tested it on a rat and a chicken and a fish, that's it. He or she has done what they're supposed to do by law. They then get their approval. The pesticide like glyphosate, which is the most used pesticide in the world, or herbicide used in the world today, they've made billions from it. And it's only now that we're beginning to realize the damage, not only to the environment, but also to our health. So again, you can see why the chemicals industry and the pharmaceutical industry don't want to give up the animal model, don't want to give up animal testing, because it's such a valuable alibi.
Okay, I'm looking at the time. We have, it was coming up quite nicely. We have one last question. That BSE was mentioned in your talk, and the question is, has that been eradicated now? And do humans still catch CJD from eating beef?
I think, well, I think CJD is more what you get from eating sheep's brains. I don't know if it's from beef, but I think a lot of it depends on whether governments have outlawed the practice. So as long as the practice is still there, because as I mentioned earlier, the mammals that have been studied all have the genes in their DNA to manufacture prion protein. We as humans also have it. Okay, we also have on our chromosome 20 the gene to manufacture the protein to make prions. So the prions are out there. Okay, we should not eat sheep's brains. We should not eat monkey's brains. We should not eat cow's brains or cow's spinal cords, etc., etc. So yes, a very good way to prevent yourself getting these exotic diseases is to certainly cut out meat and meat products, etc., etc.
Great. Okay. Thank you, Andre. I think we've covered all the questions now. Is there anything else you wanted to say to sum up?
Well, I just want to thank you and Richard very much for helping to host this webinar, this first webinar, which I think went very well. And also thank very much the participants who took the time to join us. And I'm going to now stop the recording.